Hi everyone, this is chapter 6. We're taking a look at metabolism. And just a disclaimer, the beginning of this chapter is pretty heavy on physicsy, chemistry type nebulous stuff. Okay, so if chemistry and physics isn't really your cup of tea, this is going to be a bit of a challenge just at the beginning here, but I promise we come back around to more biologically relevant topics. But it's important we talk about this idea of metabolism. Metabolism is all of the energy that is going on, right? So all of the things that are happening in the cell. There's lots of chemical reactions in the cell. A chemical reaction is one where you have reactants of some kind. They're going to undergo some type of change and they're going to produce products, okay? And this requires energy to make this happen. Sometimes when this happens, energy is released. Sometimes when it happens, energy is consumed. So when you take into account all of the different types of reactions happening and you come up with one idea about what's going on with the energy, that's called the metabolism. So it's kind of like the sum of all of the chemical reactions in the body. All right, so first we have to start with this idea of energy. Now, energy is kind of a mysterious thing. It's kind of hard to know exactly what it is in a fundamental sense. But we won't go into that type of detail. What I do want to show is that there's kind of a flow of energy, a continual flow into living systems. And here's kind of how it works. First, pretty much all of the energy that sustains you and I and life on our planet originates from energy that's produced in the sun. So there's all sorts of really interesting reactions happening in the sun that create lots of heat and thermal energy that goes throughout the universe, and a small portion of it hits our planet, and of that, some of it is escaped into heat. Not all of the solar energy that reaches our planet stays in our planet. Some of it just kind of is given off as heat. But then the stuff that does land into our planet, and specifically onto these things called plants, is what can kind of make this whole energy start being used by living systems. Okay, so there's a whole variety of organisms called producers. Producers are anything that can really do photosynthesis. Here's the equation for photosynthesis. Basically, and we'll talk more about this process later, but we're introducing it here in this chapter. Primary producers, namely plants, but also could be things like cyanobacteria and algae and things like that. They're all going to do kind of the same type of thing. They take carbon dioxide, they take some water, they take that energy that's provided from the sun, okay, and then they can actually manufacture stuff with it. So they can make what's called C6H12O6, and you should hopefully recognize this formula. It's got 6 carbons, 12 hydrogens, and 6 oxygens. The ratio here is 1 to 1. And have you seen that type of ratio before in any type of the biological molecules? And hopefully the answer is yes. This is a monosaccharide. Specifically, the monosaccharide that it is glucose. Okay, there's a couple other monosaccharides that share the same molecular formula, but this generally is glucose that's produced by plants. So they take carbon dioxide, water, and energy, make glucose, and then as a byproduct, there's a little bit of oxygen given off, which is great because that's the stuff that we need, right? So we have this really great relationship with the primary producers. So we are considered consumers, any animal that can't make its own energy, because we have to consume the energy that's made by plants. And this doesn't mean that we all just eat salads for every single meal, right? Some of us will eat meat. Well, you might say, well, hey, I'm eating meat, so I'm not really getting energy from plants. But the answer is yes, you are, because all energy comes from plants in living systems, in terms of the things that your body makes. So if you eat, for example, a cow, that cow likely was either fed corn or was fed grass, and both of those things were able to grow because of the energy they took from the sun. So whether it's directly or indirectly, we all have this special relationship to solar energy. And it's kind of weird thinking about, like, looking at your body and being like, wow, my body was like made from the sun. But in reality, in a way, it kind of was. The sun produced all of the energy of the molecules that your body uses to maintain itself and to grow. So I digress, though. the consumers are going to do is they're going to take this sugar molecule, so this is glucose, okay, so ultimately, whether directly or indirectly, they take that molecule, they add some oxygen to it, and they can then break this glucose and release the energy. So when that happens, CO2 is released, some water is made, and then this is the important thing, there's some energy released that we can then use to do stuff like make other types of molecules or grow. So this process here was called photosynthesis, spelled like this. And usually it's called photosynthesis, or it's called photosynthesis because you're making something using photons or light, photons or light. This one over here, the one that animals do, this is called respiration, specifically aerobic respiration. And respiration is also a word that's used to describe like you breathing. You like when you're respiring, it means your lungs are inflating and deflating your breathing. So oftentimes we kind of put we clarify by saying this is cellular respiration. It's not that the cells are breathing; they're making energy. So you can call it cellular respiration, and because we're using oxygen, it's called aerobic respiration as well. And the big idea is that it's the total opposite of photosynthesis. You see this? This is just a flip-flopped equation. You take this equation, you flip it around, and that's exactly what respiration is. So it's a pretty awesome system because plants will give us sugar and oxygen, and then we are going to give off, CO2 is the big thing, which then plants can then use to make more stuff. So we have this awesome relationship with the primary producers on this planet. And it's rare that you find such a perfectly symbiotic and mutually beneficial relationship in the universe, but here is one place you see it. All right, the fate of the plants and the animals after they've lived a good long life and they return to the earth, then all of the remaining energy that was trapped in the organism's body is then usually broken apart through decomposers, so bacteria and fungus and worms, and then they will release that energy as heat. One thing I'll point out is that anytime that heat is released, so like here and here, and then also there's going to be a lot of heat given off by animals throughout their life and plants throughout their life, that usually escapes and becomes an unusable form, a form that living organisms can't really use to do anything. So keep that in mind because that's going to be an important theme that we'll talk about in just a minute. When we're talking about energy, sometimes it's helpful to consider what are called the laws of thermodynamics. There's two of them that get talked about the most. The first one says something like this. There has always been, there always will be exactly the same amount of energy in the universe. So if you think about the whole grand universe, all of the galaxies and solar systems, everything, and then think about there being a specific amount of energy, and that amount will never be increased or decreased. There will never be energy made or destroyed. That's what this first law says. And it's kind of a weird thing to think about, how did that energy get there in the first place? Which is a very important question to consider, but it's one that's kind of outside the scope of this class. It's more kind of a philosophy type question. The second law is that even though there's the same amount of energy, it's changing forms. And every time it changes forms, some of the energy is that's able to do stuff is lost. So lost not in the sense that the energy is being decreased. It's just that it's a different form that can't be used to do work. It can't be used to move things, or it can't be used to make chemical bonds, or any of all the types of different stuff that living organisms would want to do with that energy. So this gets us to this idea called entropy. Entropy is a little bit more of an abstract thing to think about, but basically, to summarize it, entropy, the idea of entropy is that the world is continually getting more and more disorganized. The reason being, it takes energy that can be used to do work. It takes that type of energy to keep things organized. If you don't believe me, just consider like a bedroom of a teenager, or maybe even your bedroom, depending on how tidy you are. It takes a lot of energy to keep your bedroom clean, right? If you don't put a lot of effort into it, if you don't stop and pick stuff up and put stuff away, it just gets super cluttered and disorganized. Well, the fact that there's continually energy in the universe that's being converted to a form that can no longer be used to do work means that the universe is continually getting more and more random, more and more disorganized, and kind of cluttered, so to speak. So if you ever hear that word, that's what it means. And it's important because we're going to introduce a topic in just a minute that is going to help us keep track of how much work is going to be able to, the cells can be able to do work with, and entropy is going to be kind of involved in that discussion. All right, so here's kind of a simple scenario to kind of bring some of these more abstract ideas into an everyday circumstance. Let's say that you eat an apple for breakfast and walk to school. Okay, a very simple thing. Probably most of us have done this at some point in our life. Here's what's all going on energy standpoint. First, the apple has to be grown, and what happens there is that the leaves on the apple tree are going to absorb solar energy, and they're going to convert it to chemical energy. So the energy that's stored in all of the chemical bonds of the sugars that are inside of the apple. Well, anytime you have some sort of energy transfer, some of the energy is lost, and usually the form that it's lost in, as we said earlier, is thermal energy, so heat. So that heat escapes into the atmosphere. It kind of warms the universe up just a very small amount, and it can't really be reclaimed to do anything. Then there's the digestion of the apple. So the sugars are being broken down and they're being absorbed into your bloodstream. And this point, most of the energy is still in a chemical form, although there will be probably some energy lost through this whole process. So you might say there's some more energy lost to heat here. But the big one then is when the chemical energy in those sugar molecules is broken, and your body converts those sugar molecules into mechanical energy, which it uses for moving. This would be the energy that results from the movement of your muscles, right? So that is really what your muscles are doing. They're taking chemical bonds, breaking them, and moving little filaments in your muscles. And when you move millions of these little filaments together, it actually moves your body around. Well, this process, as you probably well know, releases a lot of heat. There's a lot of energy transfer happening here, and that's why when you're at the gym and you're working out, you break a sweat because your body's getting so hot from all that heat that's being released. So lots of thermal energy being made there. And then what you do, you could use that mechanical energy and do something with it. You could use it to climb a staircase. You could use it to do all sorts of stuff, and that's going to be an additional transfer of energy. And anytime, whatever it is that mechanical energy gets transferred into is going to release more thermal energy. And so you see that in every one of these transfers, you're losing some energy that can be used to do work. And one of the things that we like to keep track of as biologists when we're talking about chemical reactions is that amount of energy that a cell could have to do work. Okay, so anytime you have a chemical reaction, you're either going to be kind of making more what's called free energy available, or you're going to be making less free energy available. That is to say, you can either give the cell more energy to do stuff with, or take more energy from the cell so it can't do as much stuff. So Gibbs free energy helps us keep track of what's going on in that whole scenario. So what it is, Gibbs free energy, Gibbs is the name of the scientist, and then this is often just called free energy, is the energy that's associated with a chemical reaction that can be used later on. Okay, so it's kind of like the stored energy that a cell might be able to utilize to do something with. And keeping in that mind, you can classify reactions into two different types of reactions. There's exergonic reactions, and this is ones where usable energy is actually leaving. It's exiting the system. So we say that these have a negative delta G. This word, this delta, this triangle, it means change. So basically it's a negative change in free energy. So an example would be, let's say you have some reaction, there's the reactant, and then it leads to the products, right? Well, in an reaction that would have a negative delta G would mean that you have less free energy after the products are made and more free energy here, okay? So, and then in that process, there's some energy that escapes the system. So the products are going to have less free energy than the original reactants. So if you take the total free energy of the products, subtract it from the free energy of the reactants, it leads to a negative delta G. Okay, so these are types of reactions that we would say are spontaneous, meaning they can happen on their own. And this is kind of a misnomer. Spontaneous often refers to like a really, really fast event. And so these chemical reactions aren't spontaneous in that sense, but they are spontaneous in the sense that they are reactions that can happen on their own without being forced to happen. Okay. Like rust, for example, the rusting of your car. Rust just happens. It's a chemical reaction between iron and oxygen, and it forms iron oxide. And that reaction will happen whether you want it to or not, right? And so it's not like that would be an exergonic reaction because it's more of a spontaneous reaction. Generally, the series of metabolic pathways called the catabolic pathways are going to be this type of reaction, exergonic reactions. And a catabolic pathway is anything that something is being broken down. Okay. So when you have a big macromolecule that's broken down into its individual monomers, you see that there's some energy that's leaving those molecules. You know, you had a certain amount of energy stored here, and then after these are all broken down, you have a certain amount of energy stored here. But if you think about all the energy that's stored in these products, it's going to be less than the original energy in the reactants, and that's because some of the energy is escaping. And cells can use that energy probably to do some work, but then like say that these are what's remaining in the cell, well then the total free energy of these is not going to be as much as it once was, just because some of that energy is lost out of the system. Okay, so catabolic pathways are exergonic, meaning energy has left the reaction. In contrast to that, you could have an endergonic reaction. This means that energy is put into the system. So you have actually free energy entering into those reactants, and that results in what's called a positive delta G, or a positive change in the Gibbs free energy. And the types of reactions that are this sort are anabolic pathways, and anabolic pathways are different than catabolic pathways. Anabolic pathways are the series of reactions required to build stuff. Okay, and then recall that catabolic pathways are the series of reactions that will undergo, that a molecule might undergo to break it down. So an anabolic pathway might look just like the opposite of a catabolic pathway. You have a series of individual reactants, and then when you push and you put in some energy into the system, it links those together to form a product. Now, because you put some energy in, the product is actually storing more Gibbs free energy than collectively the individual initial reactants. Okay, so then there's more energy in the products and less energy in the reactants. So if you take the energy here, subtract it from the energy here, it's a positive change in Gibbs free energy. All right, and by pathway, I should make a quick note here. Pathway, what that means is it means that, you know, in this picture, we've drawn this as a simple diagram where you have these little 4 green circles attaching to one another to form this product. Well, in reality, if this was in the body, this whole event, taking the smaller things and making a bigger thing, it could require actually several separate individual chemical reactions to make the final product. So a pathway that's the definition of a pathway, anytime you have a series of chemical reactions to create the intended product. Usually the energy that we're talking about here that gets put into a cell in order to make these types of chemical reactions work is provided by breaking ATP down. And so it's kind of an interesting thing here. We have the breakdown of ATP would be a exergonic reaction because it's releasing energy. And then you have another similar chemical reaction happening right alongside of it that immediately takes that energy up and then uses it to build something. So that is an endergonic reaction. So you have the energy from an exergonic reaction being used to fuel the energy of a endergonic reaction. And this type of stuff is happening all the time in the cell. And so if you take kind of a grand summation or a grand calculation of all of the energy transfers in the cell and what is truly happening in the end, we call that metabolism. So metabolism would be like if I added up all of the chemical reactions, all of the ones that are endergonic and all the ones that are exergonic, how much energy are we really putting into the system or losing out of the system. That's the word metabolism. It refers to all of the chemical reactions going on in the cell. So like some people have a high metabolism, meaning they burn lots of energy. So they have lots of uh, need for energy to be put into the system because they burn it really fast. Other people have a slower metabolism, meaning that they don't need as much energy. They don't need to consume as much energy because their body won't use it as fast. All right, so review question number 1 then. Here's a couple of things to think about. What is metabolism? Describe the 2 laws of thermodynamics. Summarize what happens during cellular respiration. Is this anabolic or catabolic? Summarize what happens during photosynthesis. Is this anabolic or catabolic? And then where you have a product where molecules have less free energy than the reactants, what would that be? Okay, go ahead and pause the video and take a minute to work through these. Okay, here are the answers. Metabolism, again, this is the sum of all the chemical reactions happening in the body. There's 2 laws of thermodynamics. There is the first one, which says that energy cannot be made or destroyed. It merely transfers into different types of forms, whether it be chemical, solar, mechanical, like we said. The second law then states that every time you have one of those energy transfers, it's going to result with energy lost. I shouldn't say lost, that's kind of a misnomer, but energy basically, let's say the best word to say this would be something like makes energy that is, no, I shouldn't say makes either, because that makes you think that energy is being added or subtracted. What I'm trying to say is that the energy is turned into a form that's more unusable. So every time you have an energy transfer, there is energy that has become unusable. The energy is still there, it's just unusable. Okay, so if you can write that down, that's probably the way to think about it. Okay, what happens during cellular respiration? This is where we take a good old-fashioned molecule of glucose, which is C6H12O6. You add a little oxygen to it, and you end up with a little energy. You also end up with some water. You also end up with some CO2 being made. Okay, so that's taking a molecule of glucose, breaking it down. Anytime you break down something, that's a catabolic pathway. So that would be the answer there. And then what happens in photosynthesis? Well, this is just the opposite. You take a little water, you add a little CO2 to it, then you put some energy into that system, and then voila, you end up with some sugar and also some oxygen being produced. Okay, this would be an anabolic pathway because you're building something. Okay, the next question then would be: is this an endergonic or an exergonic reaction? And because you're putting energy into that system, this would be considered an endergonic one, whereas this one up here would be an exergonic. Okay, lastly, a reaction where the products have less free energy than the reactants. Okay, so here we've got reactants, here we've got products, and it's saying that the products have less free energy and the reactants have more free energy. So if you took something that was less and subtracted it from something that was more, that would be a negative change in the Gibbs free energy. So that means that energy is being lost out of this system. So that would be an exergonic reaction. Okay. All right, good. All right, so now we're going to transition to talking about some things that aren't so physics-based. They get a little bit more to what's going on in the cell. The first thing is an idea what's known as chemical equilibrium. So it's kind of a little bit of a tricky thing to think about the chemical reactions in a cell, because in reality it's not as simple as I've just shown you in these pictures, where you have, right here, this big thing being broken down into small things, and these little small things being broken down into big things. In reality, reactions can often occur in both ways. They could go in both directions. So for example, you could have a couple of monomers of some biological molecules. So this could be, if this was a carbohydrate, these would be monosaccharides. If this was an amino acid, they'd be, or if this was a protein, they'd be amino acids. If it was a DNA molecule, these would be nucleotides. All right. So you can have the monomers. You put some energy into that system, and it can create the polymer. Okay. So this is proceeding this way, but then maybe a minute or two later, this thing can then actually break right back down into its original product. So it can come right back the other way. Okay. So oftentimes you'll see this symbol right here. That tells you that there's the reaction can proceed in either direction. Either the monomers can link up to form the polymer, or the polymers can break down to form the monomers again. And how it happens depends on what, a whole bunch of things. But generally, like if there are a whole bunch of these around, it will drive the reaction this way. Contrast that, if there are a whole bunch of these around, that an unnecessary amount of them, then it will drive the reaction this way. Okay, so that's what we mean by chemical equilibrium. And if something does reach chemical equilibrium, it means that the rate at which these are being formed is exactly the rate at which they're being unformed. And so this is generally something that does not happen really in the cell, because the cell is always making a net production of something or a net destruction of something. If cells ever did reach chemical equilibrium, chemical equilibrium can happen in test tubes, and it can happen in little small parts of the cell, but if the whole cell as a whole was at chemical equilibrium, it would mean nothing was ever really in a net sense being created or destroyed. But that's certainly not true, because cells are always making and breaking stuff down. And so it's kind of an interesting thing to think about for there. Okay. All right, another big topic here that we're going to spend quite a bit of time talking about is this thing known as activation energy. So in order to describe this, consider these two reactions here, exergonic, and then endergonic is going to be the one that's over here. But we'll talk about exergonic first. Okay, and exergonic is again the one in which the delta G is negative, so it's less than 0, so that means that energy is exiting the system, as this name suggests, exert. And this is a reaction that's considered spontaneous. Okay, we said that earlier. It can happen. So for example, your car rusting, that's going to happen on its own. You don't have to force it to happen. But this is a little bit of a misnomer to say this, because it does, even spontaneous reactions need a little bit of a jump start to get them going. Okay, so for example, here's if you were to chart out the reactants and the products of a chemical reaction. So here are the reactants and here are the products. And this is the amount of Gibbs free energy, and this is as the reaction proceeds through time. Okay, so let's say you tally up all of the Gibbs free energy in all of the reactants, and it's some amount right here. Okay, and this could have a specified amount with the unit joules per mole or something like that, but I'm not worried about the actual unit of this in this chapter. So you have a certain amount of energy. And then after the reaction proceeds, the products are going to have a less amount of energy. So energy ultimately is released, because this is, the energy from that you started with and the energy that you end up with. That means the energy level of is gone down, so that means energy has escaped this system or exited this system. Well, this will happen on its own, but in order to get this reaction to occur, sometimes you have to put a little bit of energy into it. This is referred to as the activation energy. Okay, any energy you have to put into a reaction to make it occur is called activation energy. Now this is the reason why chemical reactions will require activation energy is because generally the reactants are going to have some sort of shape to them, right? The atoms are going to be bound in some sort of way to one another, and they kind of are just content to hang out and stay in that shape or stay in that form. Well, sometimes in order for you to get them to do something, you have to give them some energy so that they reach what's called a transition state, meaning the molecule is bent in some way. It changes its shape or its form in some way that then allows the reaction to proceed. So a transition state, if you ever see this word, it means kind of a shape change in the molecule or the reactant that needs to happen before the reaction, the total chemical reaction, can take place. Oftentimes the thing that supplies this activation energy is going to be heat. So like in organic chemistry, this is done all of the time, where you take a beaker of reactants, you heat them up, you heat them up, you heat them up, you heat them up, and then at some point the molecules are going to bend a certain way that leads to the reaction to proceed, and then the reactants will be converted into the products. Okay. But as I'm going to show you in just a minute here, the heat that's necessary to provide this activation energy is not produced by cells. So cells cannot produce that kind of heat required to make most chemical reactions go. You know, like if this was a chemistry test tube, you would be putting that on a Bunsen burner and boiling it and boiling it and boiling it until it finally reached its transition state and the reaction proceeded. But cells can't just like boil their molecules to make the reactions happen. So there's got to be some other way that these reactions occur, and I'll tell you a little bit. I'll tell you in a minute about how that works. Okay, on the flip side then, endergonic reactions. This is a reaction that doesn't just happen on its own. You really have to push it to happen. So these are going to be ones where the reactions start off with a certain Gibbs free energy, and you have to continually put energy into this reaction. So you have to add. Oftentimes, again, the thing that's supplying this energy is the breakdown of ATP. It's going to release energy that can push these reactions to happen. But then even though you still you can get to the energy that the products have, you still have to go over and make that transition state happen before the reaction will proceed and the products will occur. So again, this energy that you have to put in above and beyond what the final energy of the products would be, this is called the activation energy. Okay. Now, here's what I was talking about earlier. In non-biological systems, so let's say like a chemistry test tube, the activation energy is provided by heating. So you boil the heck out of that test tube until the reaction happens. But cells can't just like have little Bunsen burners in their cell and boil chemicals to make reactions happen. So, unfortunately, they don't have enough heat required to produce that activation energy. But actually, this is something that is very good for cells, even though I said this unfortunately. Because what this means is that it keeps cells from spontaneously disintegrating, right? Imagine, for example, I had, let's say I've got starch, okay? So this is a nice molecule of starch. Well, starch is made out, hopefully you know this by now. If you don't, then maybe you got to kind of go back and do some review. Starch is made out of a whole bunch of molecules of glucose linked together, okay? And generally, there's going to be a lot of free energy here. So there'll be more free energy here. And then if this reaction proceeded where all of these were broken down into individual pieces, this would be less free energy. Oops. Okay, so this is a spontaneous reaction. The starch on its own does kind of have a tendency to want to break down into its individual monomers, just because that's the tendency that things want to do. They want to increase the entropy in the universe. They want to go from high Gibbs energy states to lower Gibbs energy states and then release some energy into the world that will forever be unusable. Okay, that's just kind of the tendency of chemical reactions. But it's a really good thing for us that starch, this molecule, doesn't just automatically break down all the time, because sometimes your cells need to keep starch. They don't want to have starch just break down all the time, and then, or even all of you, let's say that this wasn't starch and this was a protein that we were talking about, and then these were all amino acids. Well, you don't want all of your proteins to just like break apart into the amino acids, because you need to have these big, large macromolecules, okay? So it's a good thing that this activation energy is required, because if it wasn't, then your starch molecules, your proteins, your big DNA molecules, these big macromolecules with a lot of free energy would automatically just break down without any restraint, okay? And then you wouldn't be able to keep your cell intact. So On one side, this is a good thing because we do not have enough heat in our cells to provide that activation energy. But then the question remains, well, then how do any of these chemical reactions ever occur? I've just told you that for a chemical reaction to occur, it requires a whole bunch of activation energy, and cells don't have the ability to reach that activation energy. Well, the solution, the grand solution, is that cells have these things called catalysts. And what a catalyst does is it actually can lower the activation energy. Okay, so here's an example. A reaction that didn't have a catalyst, it would require this amount of activation energy right here to make that reaction occur. But if you add a little catalyst in there, it lowers this down so that suddenly perhaps the cell does have just enough activation energy to make the reaction occur. The big catalyst that you see a lot of times in cells is there's going to be this thing called an enzyme. And so that's going to be what we spend pretty much the majority of this lecture discussing. But before we do that, let's quick review some of these topics. Review question number 2 now. Describe what activation energy is. How would a cell's metabolism be affected if there was no activation energy required for chemical reactions? And what effect, I should say, what effect does a catalyst have on activation energy, and why is this important? Consider these. See if you can answer these without using your notes, and then pause the video and give it a try, and then I'll go through them in just a minute. All right, so activation energy. This is going to be energy required to make a reaction happen. Or make a reaction occur. Okay. How would a cell's metabolism be affected if there was no activation energy required? Well, if there was no activation required for chemical reactions to occur, then chemical reactions would just be occurring all of the time. Anytime there was any reactant that had more Gibbs free energy than the product, it would just disintegrate, and all that Gibbs free energy would be released into entropy, further entropy. So the fact that we have activation energy makes it so that our cells, our big proteins and our DNA molecules and our starch molecules, these molecules that have a lot of free energy, it allows them to stay intact without disintegrating. Okay, so that's kind of a little bit of the answer I was looking for there. And then what would be the effect that a catalyst would have on an activation energy? These are going to lower the activation energy. And that's important because cells do not have enough activation energy to make chemical reactions happen. And so when a cell can employ a catalyst to lower that activation energy, then it means that the cell finally has the ability to have that chemical reaction proceed. And furthermore, when you use something like an enzyme as a catalyst, the cell can kind of control when and where it wants chemical reactions to happen, and that's a very important component of what it means to be alive. In a molecular sense, your cell has to be able to control its environment, and one of the ways that it controls its environment is that it has all sorts of little chemical reaction controllers. It can determine when it wants certain chemical reactions to happen and when it does not want certain chemical reactions to happen, regardless of what the tendency is going to be for those reactions in terms of when, what they want to do. Okay, so let's talk a little bit about enzymes, because these are important. Okay, enzymes are proteins. Hopefully this is kind of a review from chapter 3. And they're also organic catalysts. So there are things that the cell utilizes to bring down the activation energy of reactions and allow those reactions to occur. Now, the shape and the function of an enzyme is best described, at least up to this point, by a model that we have called the induced fit model. We can't actually go in and see what an enzyme is doing, like an animation would have us believe. But based upon a lot of data, we can come up with a model that explains kind of the trends of data that we see and how these things behave. So up to this point, this is the model that we have for enzymes. We believe that the enzyme looks something like this. It's a protein, and it's going to have a certain shape to it that is called the active site. And the active site is usually going to be a shape that fits a particular something. Okay, so the particular something that fits in there, we call it a substrate. And so in a kind of a, when you're in a chemical reaction sense, this would be the reactant. Okay, but when a reactant binds to an active site of an enzyme for whatever reason, we call that thing, though it's known as the substrate. All right, so then this goes into the active site, and then generally what happens is as it binds, there's going to be some sort of shape change that happens. Okay, so once this is an enzyme substrate complex, meaning these are kind of fused together, this enzyme will change its shape just a little bit. And then when that changes its shape, it usually does something, and oftentimes it will actually break apart the molecule. It doesn't always have to do that, though. Sometimes it could put a molecule together. Other times it could add like something onto the molecule. But in this particular picture, we're seeing that it's actually breaking that molecule into two separate pieces. And then once that has been broken, the enzyme returns its shape, which then is going to allow the products to escape and leave. So that's kind of the model of how enzymes work, at least how we understand. Now, the cool thing about cells is that they can turn on enzymes and then they can turn enzymes off. So you have this really, really precisely regulated array of chemical reactions happening. You can almost think of your cell as being like a huge organic chemistry lab where each little, there's millions and millions of little Bunsen burners in or I shouldn't say that, but there's like millions and millions of little chemical reactions happening, and the cell can tell the chemist when to turn on the chemical reactions and when to turn them off. And the way that it's done is by regulating enzymes. There's a couple ways that this is done. The first is called competitive inhibition, and this is when you have, here's our induced fit model. You have an enzyme, and the substrate will bind to it. Okay, well, sometimes there can be what are called analogs. And analogs are going to be something that have maybe kind of a similar shape as the substrate, so maybe like something like this, okay? And that analog can actually compete for the active site of the enzyme. And if the analog binds to the active site, then the substrate's not going to be able to bind. So these are called competitive inhibition. Anytime there's an analog that's going to occupy the active site, it's going to keep that enzyme from binding to the substrate. So sometimes the cell can make these analogs. Sometimes these analogs are just kind of naturally out there. But let's say if the cell wanted to turn off some sort of chemical reaction, it could produce some of those analogs, which then block the enzymes. Another type would be allosteric regulation. And allosteric regulation refers to what's known as the allosteric site. The allosteric site is a place on the enzyme that's other than the active site. Okay, so if we look, here's an enzyme. Here's the active site, the place that's going to bind the substrate. And then you see there's like a little region on the enzyme that's not the active site. Well, this is called the allosteric site. And that can sometimes bind inhibitors. So an inhibitor could attach there, and then when that happens, it makes the enzyme change its shape so that suddenly the substrate cannot bind. The function of enzymes is really determined by its shape. So if the shape of the active site is changed by an inhibitor, then it will no longer work. On the flip side, you could also have something where the allosteric site is utilized to activate an enzyme. So in this case, the natural state of the enzyme is to not be working, and then the particle in this case would be called an activator. It can bind to the allosteric site and then turn the enzyme on. Okay, so this would be like turning an enzyme off, and over here this would be like turning an enzyme on. Again, it has to do with this site here. And because that's an allosteric site that's different than the active site, this is considered non-competitive inhibition, because the thing that's attaching and turning on and off the enzyme is not actually going into the active site and competing for the active site, okay, with the substrate. So that's allosteric. And a lot of the medicine that we produce in modern medicine are going to work through either of these two ways. They are either a series of these inhibitors and activators that turn enzymes on and off. If you can turn enzymes on and off, you can control the chemical reactions in a cell. Or they could be like analogs that will outcompete enzymes for the active site. So, for example, there's a drug out there called I think this is how it's spelled. I don't know for sure, but Lipitor. And Lipitor was a drug that's prescribed for high cholesterol. And what it does is it actually shuts off the enzyme that makes cholesterol in the cell. So there's some sort of enzyme in the cell that produces that cholesterol, and Lipitor is going to be one of these things here, inhibitors or activators. Or it's going to be an inhibitor that shuts off the enzyme that makes cholesterol. Okay, so that's an example. And sometimes it's hard to figure out how many of these enzymes to shut off. Like, for example, in the cholesterol scenario that I just described, cholesterol is definitely an important part of cells, and so you want to have cholesterol around. But if you have high cholesterol, that's a bad thing too. So the trick is then to figure out the right dosage of just enough pills to take to shut off just enough enzymes so that you don't have high cholesterol, and you also still are making enough cholesterol. And that can be a tricky thing, and that's where a lot of medicines have side effects, because you start shutting off enzymes, and sometimes it can have other consequences in the cell. Okay, other things that can help regulate enzymes are these things called coenzymes and cofactors. What a coenzyme is, this is like an organic molecule, and so the dietary vitamins are this type of molecule. They have kind of a ring structure, and they can have lots of carbons and hydrogens and things like that. And these are things that actually attach to enzymes or associate with enzymes and help them run. So some enzymes actually need like a little sidekick almost. They need a little certain something to attach to them to get them going. And there's a lot of dietary vitamins that are coenzymes. For example, vitamin C is going to be a coenzyme for one of the enzymes that makes the fiber collagen. Collagen is a fiber that's found in connective tissues in the body. So like your tendons, the things that attach your muscles to the bones, the connective tissue that wraps around all of your organs and keeps them anchored, all that stuff has collagen in them. And there's an enzyme that makes this product. Okay, so it oversees the chemical reaction that makes this product. And vitamin C is one of the coenzymes that's required to make that enzyme produce collagen. And way back in the olden days, when people were traveling across the open ocean on these big sailboats, they would go for months at sea without having any type of vitamin C. And so what would happen in these long durations is that their enzymes that are used for making collagen would not work anymore. And so then they wouldn't be able to make collagen, and then they would just have all these crazy symptoms. And we called this scurvy. So one of the solutions to scurvy was to just bring some oranges along the voyage, because there's vitamin C in there, and that keeps those enzymes making collagen. Interesting example there. What a cofactor is, it's kind of the same thing. It's a little something that the enzyme needs to work, but rather than being an organic molecule, such as these big honking things that you see here, cofactors are just like ions usually, and usually like metal ions. So they're really small, just like one, atoms on the scale of just individual atoms. An example here is DNA polymerase is the enzyme that makes and builds DNA molecules, and it requires this metal ion of zinc to function. So that would be a cofactor, whereas a vitamin would be a coenzyme. The last type of regulation is feedback inhibition. And how this works is that let's say there's some original kind of substrate of some kind, and let's say it binds an enzyme, okay? And then this enzyme does something to the substrate. So let's say it changes it into this thing here, which we could call an intermediate substrate. Then let's say that this gets bound to another enzyme, and then that changes shape and it forms this thing, which is kind of a little bit different. This is another intermediate substrate. And then this goes and binds a different enzyme, which has a special affinity for this particular shape. And when that happens, it changes its shape once again, and then you have the final end product. Well, oftentimes how enzymes are regulated is in this fashion, where the final end product that's produced through this long series of reactions, which we call a metabolic pathway, because it's kind of requires several steps. Okay, so this is a pathway to get this thing all the way to this thing. It requires several steps. Well, once this thing is made, it oftentimes will go back and attach to the initial enzyme and shut it off. Okay, so you might see it going back and then changing the shape of, let's say, the first enzyme so that the substrate can no longer bind. And that's good because it means that you can't have a reaction proceeding too fast. So in our infamous example, let's say, of glycogen. Okay, glycogen is a polysaccharide of a lot of different glucose molecules. And there is a lot of this glycogen sitting around in your liver and also in your muscle cells. And when your body needs to get some energy, it's going to undergo a reaction to break this down into the individual sugar molecules, okay? And then your body can use these to make energy. Now, it would be a bad thing if your cell just like pushed a button and all of a sudden all of the all the enzymes turned on and suddenly all of the glycogen converted to glucose, because what would happen is you would have such a huge blood sugar spike you'd probably just keel over and die in an instant, okay? So your cells are highly regulated. What that means is that, let's say you need a little bit of sugar, the enzyme is going to turn on and the glycogen is going to break down. But as soon as these are starting to accumulate, they're going to go back and they're going to turn off the enzyme that was responsible for breaking this down. So you have a really nice moderation there. You can produce some sugar, but it doesn't just like rapidly break down all of your glycogen and make your blood sugar spike like crazy. Okay? And so it truly is extraordinary when you start thinking about the complexity of all of these regulatory mechanisms. You know, your cells know exactly when and where and how much of something that needs to be made, how much of something needs to be broken down. And if you look at your hand and look at one tiny little microscopic cell that you can't even see, in that one cell there are literally millions of reactions happening right now. Right in this very second, a million reactions probably happen. And your cell can start them, keep them going for a while, and then stop them, and then start them again, and then stop them. It just knows exactly how much of everything it needs to maintain homeostasis. So whenever I consider that, that's one cell, and you and I are made up of trillions of cells. So just think about how many chemical reactions are being governed right now in your body. Nothing short of a miracle. Here's review question number 3. Describe the induced fit model of an enzyme. List and describe 4 ways by which the function of enzymes can be regulated. Go ahead and pause the video and work on these, and I'll go through it. Okay, so the answers to these would be: induced fit means that you have some sort of enzyme. Here we go. Here's my enzyme. And there's some sort of active site. And the active site has some sort of shape which is going to allow some sort of substrate to bind. And when it does, the shape of the enzyme changes and it does something. If you could describe that, you have just described the induced fit model for enzymes. Okay, now the 4 ways that the enzymes are regulated. Here we go. 1, 2, 3, and 4. The first way, oh gosh, let's see if I can even remember. What was it that we talked about? Oh yeah, competitive. The next one was allosteric. Then we had these things called coenzymes and cofactors. And then the last one was feedback. Competitive means that you have some sort of enzyme with an active site, and that there's some sort of molecule. Let's say that this is the substrate, and it normally binds right here into the active site. Well, then this one right here we would call the analog, because it's not specifically meant to be in that enzyme, but it can actually go ahead and block that active site so that the enzyme no longer does what it needs to do. Okay, so that is called competitive inhibition. It's a way to turn enzymes down a little bit, make it so that they're not functioning as good as they normally would have. And depending on how many analogs are released, depends on the kind of the strength or the intensity with which that kind of inhibition is working. All right, the next is allosteric. Again, this is where we have an enzyme again. Here's our little active site. And then somewhere on the key here is that somewhere on a different spot of the enzyme, there is a little allosteric site that's separate from the active site. And into that little allosteric site, you can have either inhibitors, or you can have things that are activators binding there. If it was something that was an inhibitor, this would be turning the enzyme off. If it was something that was an activator, it would be turning the enzyme on. Okay, so a couple of different ways you could do that. So that's allosteric. And then you could have coenzymes and cofactors. Coenzymes, these are kind of organic molecules, and oftentimes these are vitamins. The different vitamins that we consume and need are the reason why we need them is because they are important coenzymes. And then cofactors, these are like our these are like our minerals. So different metal ions like zinc, magnesium, iron. These are all usually pretty common cofactors that are little ions that are required to make the enzyme work. Okay. And then lastly, we have feedback inhibition. And again, what feedback inhibition is, I'm not going to draw that because that would take me forever. But what it is that, let's say you have a metabolic pathway. You have some substrate like this that needs to somehow get converted into two separate little parts. Well, this initial substrate has to go through a series of reactions requiring a series of enzymes to get to the final product. And once the final product is made, it's actually going to work by binding to the original enzyme and shutting off that whole process. So it keeps too much of something from being made when and keeps that regulated. Okay. All right, there you have it. A little bit about enzymes, a little bit about energy, a little bit about metabolism. Again, some of this stuff can take a little bit of time to work through and process. So make sure you give yourself plenty of time to work on this chapter.